So here we start with the emerging threats, which in the microbiology lab, most of our emerging threats are really those things that develop antibiotic resistance. So we're going to go through some of the mechanisms of antibiotic resistance and those organisms that we really need to be on a lookout for in the microbiology lab. So the mechanisms of resistance are, there are numerous, but just to go through a few here. Obviously, you have enzymatic inhibition, and just a moment, not every organism will have all or any of these particular mechanisms of inhibition. The issue comes up when we have organisms that have multiple mechanisms of resistance against multiple drugs, and those are what the news likes to call superbugs. So enzymatic inhibition, so the organism produces some enzyme that is able to neutralize the antibiotic that we're using directed against that. Classic example of that are the beta-lactamases. So the beta-lactamases actually work against the beta-lactam ring in any antibiotic that has a beta-lactam ring, and there are many. And so by cleaving that ring, the antibiotic becomes useless. So if the organism has that beta-lactamase, we want to know about it in advance so that we don't start trying to treat the organism with that. And there are several kinds of beta-lactamases. There's simple beta-lactamases. There's very complex ones like the metallo beta-lactamases of CRES. In addition, some organisms, instead of worrying about the drugs that target the cell wall, they accept that the cell wall is going to be targeted, and they just produce really, really thick cell wall so that it's ineffective. Or there could be changes in the membranes of the bacterium where you can increase, for example, over here in this case, where did my cursor go? In this case here, the organism, oh, that's a terrible arrow. Let me try that again. In this case here, the organism produced coded for an efflux pump, which actively pumps the offending drug out of the bacterium, protecting it. From that, there are some organisms that do have codes for pumps that are able to do that. And they can also see changes in the cell wall itself, where the penicillin binding proteins change, so that the antibiotic can't bind. It doesn't cleave the ring on the penicillin or whatever it is, but it does. It changes the binding protein so that the drug can't bind and therefore doesn't work. Okay. Those changes in the bacterial membranes frequently are for the promotion of antibiotic efflux. If the bacteria is able, it will actively pump that antibiotic out. And then there's also the alteration of protein targets. One of the major targets, obviously one of the major targets is the cell wall. So that's where if you change that penicillin binding protein, just minor changes in that, and the penicillin won't be able to bind, obviously. And so that ends the function of that particular antibiotic. Another one is the ribosome. We know that we have several antibiotics that target either the large or the small subunit of the ribosome. It really wouldn't take much to change that particular sequence so that the drug no longer can bind to the ribosome. And therefore the ribosomes work fine. And then there are other critical enzymes that are also targeted, gyrase and things like that, targeted in the in the process of cellular reproduction. If those enzymes are modified even slightly, just slight changes in the sequence of the messenger RNA, and you end up by losing the target binding spot for that particular drug. And then finally, you also have drugs that are able to completely bypass antibiotic action. So probably the example I'm thinking of right now is vancomycin. Vancomycin kind of targets the cell wall, and it does that basically by getting in the way. So it prevents cell wall production. Vancomycin is a very big molecule, and it kind of sits down there amongst all that peptidoglycan and prevents any cross-linkings from occurring. No cross-links, no, cross-links require that the peptidoglycan be close together. No cross-links, no cell wall, bug dies. But in some cases, some examples of bacterial responses to vancomycin, where if this is where your cell wall is, normally you have the peptidoglycan trying to create a cross-link, and vancomycin sits down in there, and you can't get a cross-link, so then it just gives up and dies. Other organisms just keep building up the wall over here on either side. They don't get a cross-link until they get to here, and then you just sort of have this hole. It works. You get vancomycin resistance. Okay, so there are bugs that are able to completely bypass their action. The key thing here is, why are organisms able to develop this resistance so quickly? Well, it's because we're measuring time as humans, and for bacteria, time is much different. Okay, remember, how do these change? How does an organism learn to just keep building around without the crosslinks around vancomycin? How does an organism develop an enzyme that's capable of neutralizing another antibiotic or so-and-so? Well, the reality is it comes from genetic variability. Okay, remember that these organisms have a single circular chromosome. And that chromosome is replicated not in the order of the years, like that organism replicates itself, produces an offspring that even through asexual reproduction does still have some slight differences, mostly through point mutations. They do that in the order of minutes. We measure their new generation on the order of minutes. It's not like us where we measure generations in 10 or 15 years. We measure bacterial generations in minutes, and those point mutations can make significant differences in the abilities of that particular bacteria. And literally in a population, minutes later, you can have some members of that population that are different enough to survive a particular antibiotic assault. Okay, point mutations are one thing. You also have the regular macroevolutionary change that can occur, the forces of selection that can occur, which I tried to show here. So if this is your original population up here, they're all the same bacteria, slightly different sequences, right? There have been point mutations. This is what I'm going to call it here. There have been point mutations in these slightly different sequences. Depending on the forces, and that's these arrows here, depending on the selection forces directed against this colony, you may end up with a different resulting population, dynamic in a few literally in a few hours. So you can see here, if your forces are directed in favor of, and this is the gray arrows, so your selection forces are selecting for the gray organisms, the other organisms will tend to decrease in population, if not completely disappear. You can see in the selection of the brown organisms, we've seen definitely the little gray organisms are completely gone, and we have an extreme decrease in the beige organisms, and so on down the line. Okay, so it depends on what selection forces are there. The selection forces can be anything: environmental conditions, CO2, oxygen, the other gases that are present. The selection forces could include things like the nutritional things that are available, and they could also include antibiotics. You know, a particular antibiotic may work better against the brown population versus the gray population. Who knows? Okay. And those things are going to happen anyway. But if we throw in the acquisition of foreign DNA through plasmids, then we're talking about significant change, rapid change, where over here, if we include that, we might get a purple, or in this case, a red organism that's completely different from the original organisms. So we can get significant change if we're talking about the acquisition of foreign DNA. And you remember, how does the organism do that? So this is where a plasmid, which is a separate circle of DNA, of genetic material, can be transferred via a pilus, a sexual pilus, which allows for the transfer of this material from one organism to another. Most of the time that occurs within a species, but it can occur across species. So what are the risk factors for these kind of stresses that select for the more resistant organisms? Well, one of the big things is when you take sick people and you put them together. Okay, so the risk factors here have to do with lots of people close together. Okay, so you can see in the daycare center, obviously, oops, where are we? We have crowding. In hospitals, we have clusters of people. Um, and in a long-term care facility, we're keeping all the patients together, same patients. And then we're going to throw in animal feedlots sort of as a different group, okay? But for the most part, medically, daycare centers, hospitals, and long-term care facilities. So old folks home, rehabilitation home, any kind of long-term care facility, they all have their risk associated with them. Okay, and you can see what's the big deal in each of these. Well, daycare centers is filled with children who are by definition immunologically naive. Okay, if they're immunologically naive, that means they have to get infections before their immune system responds to them. So they get a lot of infections, and they're dirty little things. They're messy children, and so the children, and unfortunately sometimes the staff, do not actually clean their hands, clean their environment enough in order to prevent the spread of these viruses from one person to the other. And then add on top of that the fact that at least some of the population in a daycare are wearing diapers, so that they're not, they have urinary and fecal incontinence because they haven't been trained yet. So a staff member changes a diaper and doesn't properly wash their hands and then goes and cares for another child, and you have the transmission of disease right there. Okay. In hospitals, we know we have immunocompromised patients in hospitals. When patients are in a hospital, there's something wrong. So obviously they need, we have to consider them starting out as immunocompromised. Even if they're completely healthy, they probably have a wound, maybe they had surgery or dialysis or something like that. So they're at least, they have some risk involved if they're in the hospital. In addition, they tend to be on antibiotics. Okay, so you got all these people hanging out. Antibiotics will select for resistant bugs, and then that person who's got that selection going on is sitting next to another patient who's on a different antibiotic, and you've got this commingling of high-risk individuals. And they also, in hospitals, they tend to use broad-spectrum antibiotics. So there's a lot of movement away from that. And also prophylactic antibiotics, which is where we give patients antibiotics before they ever have an infection, something that is very dangerous. Okay, and then if our staff doesn't follow proper infection control, proper standard precautions, then that's, it doesn't matter if you're in a rush, you can really change somebody's life by giving them an infection if you don't wash your dang hands. And then finally, long-term, well, not finally, but long-term care facilities. These are your nursing homes and your rehabilitation centers. Again, these patients are immunocompromised. The elderly are by definition immunocompromised, and then those who are going through rehab have had a recent traumatic event most likely, and so they're also immunocompromised. And there's a lot of they've already been on antibiotics at some point during their care most likely, and so you get these resistant microbes that these patients actually harbor. Okay. In particular, in long-term care facility, we see things like decubitus or pressure ulcers, wounds. There is frequently fecal or urinary incontinence, if not both. And so cleaning these patients and caring for them can lead to the staff transmitting the bug from one person to the other. Precautions are vital in these areas. These are very, very high risk. The long-term care facility is probably the worst of all. And then we have to add in the animal feedlots because in these, super farms kind of thing, these commercial farms, they have a tendency, they're growing up lots of animals really fast, and they have a tendency in order to get the healthiest animals to use antibiotics, which will actually promote a larger, healthier animal, but unfortunately, the bacteria that they harbor, their normal flora, which include things like Salmonella, Campylobacter, E. coli, Enterococcus, those bacteria then become resistant. So if you get Salmonella, say, from a chicken who's been on antibiotics, you know that the antibiotics that that chicken used, you can't use. Okay, so these are the things that are problem. And also, the doses that they use on animal feedlots are animal doses. So for a human, they would be considered subtherapeutic. In addition, those doses, they're even subtherapeutic for the organisms that you're targeting. So since they're really low doses, the bacteria really doesn't affect them. It really actually enhances the selection towards a resistant organism. And no matter what you do, animals do not learn to use the toilet and clean their hands. So obviously sanitation is just going to be poor by definition. There's animals there. Okay, so there's poop somewhere around. So then we end up with the people working there becoming carriers or harboring those organisms, bringing them home or into the community. So let's go through some of the mechanisms, okay? So enzymatic alteration, which was the first thing we talked about. Obviously, the first thing that we knew about, we had staph organisms. We treated them with penicillin. First thing staph did was produce beta-lactamases, which means that they probably harbored those beta-lactamases throughout history, which does make sense. The beta-lactamases that they produce, that we started with, was penicillin, which is a product of molds. Staph and molds have had to grow up together, and so they developed enzymes to protect themselves from the mold, okay? And then we have Enterococci and gram-negative bacilli. Those also have modifying enzymes, several modifying beta-lactamases, that they actually can transmit to each other via plasmid, okay? There have been at least 340, if not more, types of beta-lactamase characterized, and we know what the beta-lactamase does. It splits the beta-lactam ring of any penicillin type antibiotics. So then, I don't know, 15 years ago, 20 years ago, we characterized this new beta-lactamase called an extended spectrum beta-lactamase that actually it extends the spectrum of against which this particular beta-lactamase works, the spectrum of drugs, and limits your treatment options just to the carbapenems, which would mean like imipenem, meropenem, those ones, ertapenem. And so this was found, ESBLs were found, not in all, but in some of the specimen of the species, and you can see the genus that were implicated, Klebsiella being the biggest one. The second biggest is still going to be E. coli, most likely, but it'll include also Proteus, Pseudomonas, and Citrobacter. Those all can harbor ESBLs and likely transmit them not only between the same genus, but also from one genus to another, to a whole different organism. Okay. So ESBLs developed, and we were calling, oh my gosh, got ESBL, oh my gosh, ESBL, we were reporting it to the pharmacy, it was a big deal. And then, I could say probably 10 years ago, I guess, carbapenemases rapidly developed after the ESBLs. This is an even broader spectrum of penicillinase. It's actually a metallo-beta-lactamase. It's able to basically neutralize, well, the carbapenemase is a step below the metallo-beta-lactamases. The carbapenemase is able to neutralize even more antibiotics, your imipenem, meropenem, or dependent, they're not going to work. And so you're limited to some of the more toxic antibiotics, and they're very difficult to treat. The organisms that harbor carbopentamases, primarily Klebpneumo is the organism, but we have found carbopentamases present in E. coli, Citrobacter. So it is present in other ones, but primarily you're going to find it in Klebpneumos. If you do get a Klebpneumo from a nursing home patient or an acute care facility patient, you need to make sure that it's not carbapenemase positive. And then finally, the metallo-beta-lactamases. These guys are terrible. They're highly resistant to almost all almost all antibiotics. Pretty much all the ones that are on routine formularies, they won't work with the metallo-beta-lactamase. And you can see that it actually, this little cartoon shows you how it works. It cleaves this you see here, where it cleaves this ring here. Making the drug useless. So it really takes out that beta-lactam ring pretty well. The only drugs that have shown some activity against a metallo-beta-lactamase producing organism are the tigecyclines and colistin. Unfortunately, both of them are pretty toxic. So your patient has to be able to withstand that particular antibiotic. Okay, so now if you think about it, so who's going to get this? A patient in a long-term care facility who's potentially has things like kidney disease, liver disease, things like that. You have to worry about how colistin, how tigecycline will work on their system. So these are major factors. The carbopentamases, metallo-beta-lactamases. Carbapenemase we actually directly test for. Metallo-beta-lactamases you recognize as the entire panel comes up resistant, and you're like, oh, gotta call a doctor. Okay. At this point, CDC requires or suggests, they don't require, they suggest that we call any organism that is resistant to more than one class of antibiotic that means if they're resistant to 2 classes of antibiotics. So is it resistant to your penicillins and your cephalosporins? A lot of things are resistant to that. We're supposed to consider that a multi-drug resistant organism, and we're supposed to call the physician. So that's a lot of calling done in the micro lab. Okay, so changes in permeability. How can the organism change its membrane or cell wall permeability to resist a particular antibiotic? Well, there's a few mechanisms. Remember that the drug has to be able to get into the cell in some cases in order to act. And those are anything that's directed against a ribosome, against an enzyme, that's get into the cell, right? So gram-negatives, for example, gram-negatives are resistant to almost all the penicillins, mostly because they have a lipid bilayer. So that drug can't actually get across that the lipid bilayer. So it never gets access to the full cell wall. Doesn't work. Okay, so that's just one example. Other ones, so that in the gram-negatives, you wouldn't treat it with those penicillin drugs. It's not going to work. Okay. Other antibiotics, for example, hydrophilic antibiotics or water-soluble antibiotics, they are able to penetrate that gram-negative barrier because there are porins present in the membrane of the gram-negative, so the antibiotic makes it through. However, minor changes in the sequence or mutations of those porin genes can create resistance, like the resistance that we see in Pseudomonas aeruginosa to imipenem, for example. The resistance that Pseudomonas has against imipenem is not due to a carbapenem resistance, like we might see a carbapenemase, like we might see in other gram-negatives, but more frequently it's due to a mutation in the porins that it just doesn't allow it to get in and get full access to the gram-negative. Then we have other mechanisms of change. So in this here is an example of the gram-negative bilayer, where normally the Pseudomonas can go through this porin, but a minor change, and it can no longer go through. Aminoglycosides require active electron transport chains to gain access to the cytoplasm. So it's an active transport across the membrane. So if you see here, when aminoglycosides move through, it requires energy in the form of electrons in order to do that. So this particular transport protein requires energy. Staph is able to alter the energy generation for these proton pumps, so that they no longer use electrons. Or I'm sorry, these are proton pumps, so that they actually don't use the electrons usually required, and the aminoglycosides can't get in. So it's we used to not see this. We say we don't really see this, but we do see it more often, especially in the long-term care facilities. And in some instruments, it might say high resistance aminoglycosides noted, and you just know that it's likely a transport pump change that's occurred. The antibiotic can't make it through the membrane. And then finally, we have antibiotic efflux, something we mentioned before, and this is present. Pseudomonas and Staph are both able to actively pump out tetracycline out of their cell, the cytosol. Okay, and this ability is most effective when the doctor is using subtherapeutic concentrations of tetracycline. But it has also been associated with some quinolones and stuff. The key thing here is that adequate dosing. So this is where the antibiotic is moving in, and then the cell is turning it around and actively pumping it directly out. That'll work most of the time. All you have to do is add more and more of the drug, and the pumps won't be able to keep up. Okay, but it has to be the proper amount. Okay, how about those protein targets that we have in the cell and the cytosol? How do the organisms develop resistance against those drugs that are directed against protein targets? Well, for ribosomal binding sites, for example, the tetracyclines, the macrolides, clindamycin, aminoglycosides, these all normally target the ribosomal binding sites. So when we see resistance to those drugs, most often the cause is that the sequence on the ribosomal RNA has been slightly altered so that the antibiotic can't bind and thus can't interrupt protein synthesis. Either if the sequence has been changed and the antibiotic can't bind at all, or the sequence has been changed to accommodate the antibiotic, so it can bind all at once, but the ribosomal RNA is still able to function. So either one of those methods is at play when you see resistance to any of those drugs, or likely at play. Then we have some other cases where, like for example, ribosomal resistance is common with streptomycin, but uncommon with a drug like gentamicin and tobramycin and amikacin. Those are all related drugs. So sometimes we see the resistance occurring in one drug in a class, but not in all the others. So streptomycin is a little less useful these days. Gentamicin, tobra, and amikacin still pretty useful. They're associated with a little bit more toxicity, but they're a lot more useful than streptomycin. A good example of a completely established resistance, ribosomal resistance, is the resistance that Staph aureus and Enterococci have to macrolides. We don't even report macrolides for these two bugs because it's established that they're going to be resistant to it. Okay, so how about the cell wall precursor targets? So down here we are. The key thing there when we're talking about vancomycin, there's a couple of different mechanisms of resistance to vancomycin. We really are mostly interested in the resistance that exists with enterococci. The resistance that enterococci has to vancomycin is classified as either A, B, or C based on the level of resistance. Level A is a very high level of resistance, and it's associated with the VAN-A gene. The VAN-A gene encodes basically an inducible protein, so a protein that will only be produced in the presence of vancomycin. And it's involved in cell wall synthesis, and it's found actually a lot in E. coli, but also in enterococci. It can be found in enterococci. And basically, this gene encodes for a different amino acid sequence than normal cell wall peptidoglycan. And so you end up with pretty high resistance. It really isn't affected by vancomycin at all. So the vancomycin can't even settle in to the peptidoglycan. Then we have B resistance, which is moderate resistance and associated appropriately with the VAN-B gene. And then C resistance, which is low level resistance, which is associated with the VAN-C gene. When we screen for vancomycin resistance, we would mostly look for the VAN-A or the A resistance, that high level resistance, because we might be able to overcome B and C if we just increase the dosage of the vancomycin. Again, we have to watch the liver, you know, some of one, half the other, okay? So you can see this little cartoon over here shows you how the change in the amino acid sequence codes for, instead of a D-ALA, we get a D-LAC, and vancomycin can't bind. And you can see what vancomycin does. It just sits in the way. No peptidobinding. You can't bind any, you can't do any cross binding there. Now, every time we get a Staph aureus isolate, the idea is that we really probably should be screening all Staph aureus isolates for decreased susceptibility to vancomycin. One of the recommendations is that, in particular when you get it from a sterile site like a blood, the recommendation is to drop a vancomycin disc on a blood plate and grow up the Staph aureus around it. And you're going to look for isolates that grow within the zone of clearing of that vancomycin disc. Any isolates that grow within that zone of clearing, this is something that we used to do on blood cultures. You would need to work up further and separately from the main isolate. It's still a Staph aureus, but it has a slightly different sensitivity profile, and you have to ensure that it's not vancomycin resistant. And then some other enzymes that are present, some drugs bind, instead of directly binding to the peptidoglycan, they bind the transpeptidases, or the penicillin binding proteins, the transpeptidases that actually help to create the peptidoglycan, and alterations in those enzymes can lead to beta-lactam resistance. So here is a chart for you, a nice tidy chart that gives you the organisms, the emerging organisms that we're looking at, which class of antibiotics they are resistant to, how they're resistant, their mechanism of resistance, and if there's any chromosomal aspects that could potentially lead to a molecular screen for that particular organism. You can see here, if it's chromosomal, meaning we really haven't necessarily, or plasma mediated, we don't necessarily have one gene that we could target molecularly to screen for this. So some of the other ones, we can screen molecularly, but not all of them. So you can read through this. I'm not going to read it to you. It's all there. What's the mechanism? What's the class of drugs? You got to know this. And then finally, I put in, well, it's not finally, but I put in this chart here for you because a lot of times in microbiology, even though we deal with the drugs all the time, we don't necessarily know it from like a pharmacologist point of view. So here's the chart to let you know which drugs belong in which class. Penicillins, pardon me, at this point it's pretty much the smallest class because there's so much resistance to penicillin G. And then when you add some of the penicillinase resistant penicillins, you're still with methicillin, oxacillin, and stuff like that. The cephalosporins come in 4 generations. Resistance on one generation usually will mean resistance to that entire class, but not necessarily to the next generation. So although if it's like 3rd resistant, you're probably not going to go back to 1st. It is possible that all the generations are resistant, and then so on, fluoroquinolones, aminoglycosides, monobactams, carbapenems, macrolides, and other. This is something, there's new drugs developed all the time, and they've got to be shoved into one of these classes. Pardon me. This is something I always have to refer to. This is, I always think of this as pharmacist's job, but really, since we deal with them, it's very helpful to know the classes. And these are the chemical names. So the doctor might call up and ask you for something, even, a commercial name. And you're like, huh? So another chart that's nice to keep in the lab is also a translator between the commercial name and the chemical name, which is what we use in the lab. Okay. And then this is just another chart that groups the antibiotics by resistance mechanism. I'm sorry, by mechanism of destruction. So how do they work? So the antibiotics that target cell wall synthesis, protein synthesis, and so on down the line. You've seen this before, but just as a reminder for you to have it. Read it, you need it. And then finally, intrinsic resistance. Intrinsic resistance is important. There are certain organisms that are intrinsically resistant, I mean, built-in resistance. It didn't develop it to a particular either group or class of antibiotics, which means that you would not include these drugs in your sensitivity panel when you're considering either reporting to the doctor or whether or not it's a multi-drug resistant organism. And so you know about this, we've talked about this before, aminoglycosides do not work against anaerobic bacteria. They never have. So don't say, oh my gosh, it's suddenly resistant. They don't work, they never did, okay? You wouldn't use metronidazole against an aerobic bacteria. It doesn't work, it never did, okay? So these are the drugs that you would not include for these particular organisms, and as such, you shouldn't consider them in your sensitivity report because by including, even if it says resistant, some doctors think, well, I can make it work, so we don't include it in the sensitivity report at all. It shouldn't be.